Module F, General Adaptive Trials with Multiple Objectives. In this module, we will switch from group sequential trials to a general class of adaptive trials. The main difference between the group sequential setting considered in Module E and the setting we're going to consider in this module is that in general adaptive trials, we deal with multi-stage designs that support a potentially very large number of decisions at the end of each stage. This includes, for example, options to increase the sample size or select the best treatments to be analyzed at the final analysis, whereas in group sequential trials, only two decisions can be triggered at an interim analysis. A trial can be stopped early due to superior efficacy or due to lack of efficacy. In other words, due to futility. No other decisions are supported. Given the complexity of decision rules utilized in adaptive trials, more general principles have been developed, and most commonly the so-called combination function principle is applied in multi-stage adaptive designs. The great news is that this principle can be applied along with the closure principle, which enables us to handle several multiplicity components in a trial along with repeated significance tests at the individual decision points. In section 1, we will begin with a review of underlying principles and commonly used decision rules employed in adaptive designs. As an illustration, a case study based on an adaptive population selection design will be presented. There will be a good amount of technical details in this module, as well as various derivations, but we will stay at a fairly high conceptual level. Section F1, Decision Rules in Adaptive Designs. The general adaptive design framework, as you know, has attracted much attention in clinical trials over the past 15 to perhaps 20 years due to the appealing features of adaptive designs. Examples of data-driven adaptations that can be enabled in confirmatory trials are shown on the slide. This includes options to increase the sample size or increase the target number of events in time-to-event trials, to enable adaptive treatment selection as well as adaptive population selection at an interim analysis. And there are many other less popular options, such as switching the trial's goal from, say, a non-inferiority test to a superiority test. We should not forget that adaptive designs do not exist in a vacuum, and trials that take advantage of certain adaptive features typically pursue multiple clinical objectives that are formulated in terms of several endpoints, primary as well as secondary endpoints, potentially several dose control comparisons or several patient populations. There are maybe other sources of multiplicity. Our main goal in this module is to discuss available methods for tackling multiplicity issues in adaptive trials. A good example of an adaptive trial with additional multiplicity components is the phase 3 trial in patients with locally advanced or metastatic breast cancer that we introduced in Module A as example 5. A quick summary of this trial example is presented on the slide. This is a 2-arm trial. An experimental treatment will be compared versus control. The primary endpoint in this trial is progression-free survival, and what is more important from our perspective right now is there are two sources of multiplicity in the trial. There are two predefined patient populations and two decision points. Let's begin with the first multiplicity component with two predefined patient populations. We said back in Module A that a biomarker-driven design is employed in this trial. The selected classifier is based on a genetic marker, and at the time of enrollment, patients are classified as classifier positive if gene mutations are identified, or as classifier negative otherwise. We will also refer to this subpopulation as the target subpopulation or biomarker positive subpopulation M plus. An effectiveness claim can be formulated if a beneficial treatment effect is established within the subpopulation. This claim is typically referred to as the restricted claim, and alternatively, treatment benefit may be established in the overall trial population, which is known as the broad claim. It needs to be emphasized that the treatment effect will not be evaluated in the biomarker negative subpopulation, or M minus, as part of the primary analysis. This assessment will most likely be performed, but only as a supportive analysis or even as a purely descriptive analysis. It will have no impact on the product label. And here's the second multiplicity component in the trial. There are two decision points with a single interim analysis and the final analysis. Multiple decisions may be enabled at this interim analysis. As you can see on the slide, the sponsor may be interested in pre-specifying an early stopping rule due to futility or superior efficacy, and those rules would be applicable to both the overall population. They would depend on a strong or weak treatment effect in the overall population, as well as the biomarker positive subpopulation. The sponsor may be interested in enabling a rule to adjust the target number of events in this trial. And there's also an option, which is the last option here, to specify a population selection rule. This means that the efficacy signals within the overall population and subpopulation will be examined at the time of the interim analysis, and a decision may be made to perform an assessment of the treatment effect in both patient populations at the end of a trial. Or if the signal is much stronger in one of the populations, then this patient population may be chosen to be the main focus at the time of the final analysis. In this case, the other patient population may be demoted, so to speak, to a secondary analysis, or it may not be analyzed at all. For example, if there is no evidence of a beneficial effect within the biomarker negative subset, then enrollment of biomarker negative patients may be discontinued after the interim analysis, which means that the overall population analysis will not be even defined at the end of the trial. In this module, we will concentrate on this last decision rule, and later in this module, we will discuss multiplicity considerations that are specific to adaptive population selection designs. Adaptive design methodology. Our ultimate objective in this module is to present a class of multiplicity adjustment strategies applicable to adaptive trials with multiple objectives, and we will begin with baby steps. This section will help us prepare for defining the general methodology, and we're going to focus first on adaptive designs in a traditional setting, by which I mean that we will consider an adaptive design setting with a single source of multiplicity due to repeated significance testing of the treatment effect at two or more decision points. There will be no other sources of multiplicity. Multiple approaches have been developed in the literature to build flexible adaptive designs that support virtually arbitrary data-dependent changes at an interim analysis, and probably the most popular approach is the combination function approach that I mentioned at the beginning of the module. It is broadly used in confirmatory trials, and there have been dozens and dozens of papers that examined various aspects of the combination function approach, as well as various extensions of this approach. I will only reference here the very first paper on this topic that was published in 1994. Other approaches to setting up flexible adaptive designs that may be mentioned here include the conditional error function approach. This approach is more commonly used in the context of clinical trials with sample size adjustment rules, and it will not be discussed in this module. To define the underlying combination function principle, we will focus on two-stage settings with a single interim look, which is followed, of course, by the final look. I would like to point out that the combination function principle is not limited to simple two-stage designs. It can be applied to adaptive trials with any number of stages. It will be helpful for us to first introduce the notation that would have been used, or a general approach that would have been applied in a group sequential design with a single interim analysis. In a group sequential setting, we would compute the treatment effect statistics and p-values denoted by z1 and z2, or p1 and p2, and those test statistics or p-values would be computed at the interim analysis and at the final analysis. The key point here is that the test statistics and p-values are based on accumulated data rather than stage-wise data. For example, Z2 and P2 are derived from all data available at the final analysis. The combination function principle approaches analysis of data from multi-stage trials from a different perspective. In very simple terms, test statistics and p-values are first computed from each stage within the trial, and then they are combined across the stages using appropriate methods. To compute stage-wise test statistics and p-values, it's common to consider so-called patient-wise separation schemes. What happens here is that the trial's dataset is partitioned into two non-overlapping subsets that are referred as the stage 1 dataset and stage 2 dataset. To give you a quick example, in a trial with a fixed patient follow-up, for example, the length of the treatment period is 6 weeks for all patients, it is common to define the first stage as the set of all patients who complete the treatment period prior to the interim analysis, and then the remaining patients, which means patients who complete the trial after the interim analysis, will be included into the stage 2 dataset. But when the primary endpoint is a time to event endpoint, it is more common to draw a line between the two stages in a different way. It is accomplished by including patients who are enrolled in the trial by the interim look in stage 1, and the patients who are enrolled in the trial after the interim, they become stage 2 patients. The number of patients in the stage 1 dataset will be denoted by N1, and the number of patients included in stage 2 will be denoted by N2. In addition to this, we're going to introduce the stage-wise test statistics and p-values. We're going to use asterisks to be able to differentiate between the test statistics and p-values that are used in a group sequential testing and in a multi-stage testing. As you can see on the slide, Z1 asterisk and Z2 asterisk will denote the test statistics computed from stages 1 and 2, and P1 asterisk and P2 asterisk will compute the corresponding p-values. The reason patient-wise separation is emphasized in multi-stage trials is that it results in independent test statistics, and independence of stage-wise test statistics plays a central role in the combination function approach. Here is a simple diagram that is conceptually quite similar to diagrams that we used a lot in Module E. Here we see the time axis, or the information axis, that begins at 0, t equals 0. That indicates the beginning of the study. T equals 1, or the information fraction of 1, of course, indicates the end of the study. And the interim analysis is performed at the information fraction of t1. So as shown on the slide, all of the data that is available to us prior to the interim analysis will be referred to as stage 1, and then stage 2 captures or serves as an umbrella for all of the data that will be collected between the interim analysis and the final analysis. And this slide simply reminds us how we define the stage-wise test statistics and stage-wise p-values. They are computed, as you can see here, from each individual stage as opposed to using all data that are available at the time of the final analysis. Another important component of the combination function approach is pre-specification of stage weights. The weights will be denoted by W1 and W2, and naturally we require that the weights must be positive and they add up to 1. In general, the stage weights do not have to be proportional to the information fractions, but it's pretty common to have those weights chosen in a way that they are proportional to the information fractions. For example, as you see at the bottom of the slide, W1 would be equal then to N1 divided by the total sample size, and then W2 would be equal to N2 divided by the total sample size. The only real requirement here is that the trial sponsor must predefine the stage-specific weights. It will be helpful to compare the traditional test statistics used in group sequential trials and stage-wise test statistics that provide a foundation for the combination function principle. We can see here, if we begin with test statistics, that the two test statistics are equal to each other at the interim analysis. And then when we compare the traditional and stage-wise test statistics at the time of the final analysis, then Z2 can be shown to be simply a weighted sum of the stage-wise test statistics. The weights here are proportional to the squares of the predefined stage weights. And when we switch to the p-value scale, once again at the interim analysis, the two p-values are equal to each other. And when we proceed to the final analysis, we can show that the standard traditional p-value, denoted by p2, is actually a function of the two stage-wise p-values. And this function c is known as the combination function. Combination functions play a central role in the combination function approach. That is to be expected. And I would like to point out that multiple combination functions have been developed and are used in confirmatory trials, but most commonly it is the so-called inverse normal combination function that is used. This function is defined on this slide. It depends on the predefined weights, stage weights W1 and W2. It will be worth mentioning that using the expressions given at the bottom of the slide, that with this particular function, the combined p-value derived at the final analysis is indeed equal to the standard p-value computed from the data available at the end of the trial. Now that we have understood the relationship between the traditional group sequential approach and approaches used in general multi-stage clinical trials, it will be quite straightforward to formulate decision rules at the interim and final analyses. We're going to begin with the interim analysis using the kind of a diagram that we used in Module E. In this case, as you can see, the decision rule at the time of the interim analysis, and this decision indicates that the sponsor may be able to stop the study early due to superior efficacy because the null hypothesis of no effect is rejected. This decision is triggered by the inequality that the stage-wise p-value computed from all of the data available to the sponsor up to the interim analysis is less than or equal to U1. And of course you'll recognize U1 from Module E. This is simply the adjusted significance level at the interim analysis. This significance level can be derived from an appropriate alpha spending function, including the functions that we discussed in Module E in the context of group sequential designs. And of course, if this stage-wise p-value is greater than U1, then a decision will be made to continue to the final analysis. Now, at the final analysis, instead of using the traditional standard p-value, p1, we're going to apply the predefined combination function to the two stage-wise p-values, and we're going to compare this combined p-value with the appropriate adjusted significance level denoted by U2, and the null hypothesis of no effect will be rejected, which signifies that the treatment effect is beneficial if this combined p-value is less than or equal to U2, and otherwise the trial's outcome will be declared negative. A very important conclusion regarding the relationship between group sequential and multi-stage designs is presented on the slide. As we have seen, decision rules based on a combination function are equivalent to decision rules used in group sequential designs, provided the trial's design is not modified at the interim analysis. So we know that multi-stage designs are a simple extension of group sequential designs, and if we are not planning to update the trial's design, we are only interested in basic decision rules that support early stopping due to superior efficacy or lack of efficacy, then the two approaches are equivalent to each other. It is natural to ask, of course, the question, what happens if we decide to modify the trial's design at the interim analysis? Well, if we indeed decide to modify the trial's design at the interim analysis, for example, as you can see on the slide, the sample size may be increased, or the target number of events may be increased, or the sponsor may be interested in enabling a patient population rule, well, this is where the combination function principle really shines, because it's been shown that the combination function principle protects the overall type 1 error rate for any design adaptation, provided that the stage weights are predefined. In simple terms, all a trial sponsor needs to do is to first pre-specify the stage weights, then run the analysis, compute the stage-wise p-values, and finally apply the predefined combination function to perform inferences at the final analysis. In this case, the overall type 1 error rate will be protected. The reason we have spent so much time on defining the combination function principle and also discussing its properties is that this principle addresses a well-known problem that arises in clinical trials with adaptive or data-driven decisions. It is known that virtually all data-driven decisions applied at an interim analysis end up affecting the distribution of the stage 2 statistic, denoted by Z2 asterisk. So if we simply go ahead and analyze the data at the final analysis using the standard test statistic Z2, the overall type 1 error rate will be inflated. To control the error rate in an adaptive trial, the test statistic used at the final analysis must be properly adjusted, and this is exactly where the combination function principle comes in. The combination function approach provides such an adjustment to protect the overall type 1 error rate in an adaptive trial, and this principle is applicable to a very broad class of adaptive designs. I would like to remind you that we have assumed so far a relatively simple setting with a single source of multiplicity. We assume that multiplicity is induced by repeated significance tests at the interim and final analysis, but there are no other multiplicity components in the trial. In reality, it is increasingly common, and I've emphasized it multiple times already, to pursue multiple clinical goals in multi-stage adaptive trials, and the breast cancer clinical trial, or example 5, serves as a good example. How do we incorporate additional sources of multiplicity? That's really where we need to rely on the closure principle, or in other words, on the closed testing approach to handle those additional multiplicity components. The diagram shown in the slide helps us understand the general ideas. I know it's a busy slide, but this simply reflects the fact that multiplicity problems arising in adaptive trials with data-driven decisions are indeed quite challenging. What we see in this slide is a matrix. This matrix has three columns, and the columns correspond to stage 1 data, stage 2 data, and then final analysis, which is of course based on combining the data from the first two stages. And the rows in this matrix correspond to the null hypothesis of interest. To be able to protect the overall type 1 error rate, as I have said before, we are interested in applying simultaneously the closed testing approach and the combination function approach. The closed testing approach, as indicated on this slide, is applied across the hypothesis tested in the trial, and the combination function approach is applied across the stages of the trial. And the beauty of this approach is that it supports any data-driven adaptations. You will notice that, for example, the box corresponding to the hypothesis H2 is missing from the second column, the stage 2 column, and this is actually quite intentional. I did that to show you that based on the results of the interim analysis, a decision may be made to remove this hypothesis from consideration. For example, a certain patient population may be removed and the final analysis may be performed only with respect to the remaining null hypothesis. When we combine the two approaches, we can guarantee that even in this and more complicated types of adaptive decision rules, the overall alpha will be indeed controlled. Methods for dealing with complex multiple objectives in the context of group sequential trials or general adaptive trials have been proposed and reviewed in several recent publications. Here I will mention just a few recent publications. These papers will help you get started and learn more about this interesting class of multiplicity adjustments. And if you would like to learn even more, you will find references to other relevant papers in the papers listed on the slide. Section 2, Adaptive Population Selection Trials. In this section, we're going to apply the general ideas that we introduced earlier in this module. I will begin with a quick roadmap to develop decision rules for an adaptive population selection design that will be applied to example 5 based on the breast cancer trial. We will first define the key elements of the two-stage design that will be employed in this trial. After that, we will be ready to focus on the development of a multiplicity adjustment strategy for this trial that takes care of two sources of multiplicity. Using the ideas summarized in the diagram that I presented on slide 24, we know that to construct a method for controlling the overall type 1 error rate in this trial with complex decision rules, we will need to simultaneously and creatively apply two different principles. First, the closure principle will be applied and it will help us handle multiplicity induced by the first source. The first source here is the evaluation of treatment effects within the overall population and the pre-specified subpopulation of patients. The second source of multiplicity, due to repeated significance testing at two decision points, interim analysis and then final analysis, this source will be addressed by applying the combination function principle. We will begin by specifying the stages that will be used in this trial design, and here we will use a patient-wise separation scheme that we defined earlier in this module. Specifically, as you see on the slide, the stage 1 dataset will include all of the patients who are enrolled prior to the interim analysis, and then the rest of the patients who were enrolled after the interim analysis, will be included in stage 2. Using this information, it's quite easy to define the stage-wise test statistics and p-values. We'll use the same notation as before. Z1 asterisk and Z2 asterisk will denote the test statistics computed from the stage 1 and stage 2 datasets. Likewise, P1 asterisk and P2 asterisk will denote the p-values associated with the stage 1 and stage 2 datasets. we're going to make sure that the test statistics and p-values computed from stages 1 and 2 are independent. This is a very important fundamental assumption made by the combination function principle. We're going to spend a couple of slides discussing an important consideration related to a potential violation of the independent assumption. I have pointed out earlier that it is critical to ensure that the test statistics computed from stages 1 and 2 should be independent. By applying the patient-wise separation scheme that we defined on slide 27, we created two non-overlapping groups of patients, which means that they are independent. This is a great start, but we also need to make sure that the sets of events that occur in stages 1 and 2 are independent from each other. To help us understand what could potentially go wrong here, let's take a closer look at the censoring schemes that are applied in each trial stage. We know that when we begin with the stage 1 dataset, that primary outcomes, PFS outcomes, will be censored at the end of the trial. The same principle will of course apply to the stage 2 dataset. PFS outcomes will also be censored at the end of the trial. Now let's think about a potential impact a data-driven adjustment of the target event count could have on the censoring scheme. For example, suppose that a decision is made at the interim analysis to increase the original target event count. Would that present any problems as far as the independence assumption is concerned? If the original target event count is increased after the interim analysis, the length of event follow-up in stage 1 will depend on the data collected in stage 2. What I mean by this is that the length of follow-up for patients who are included in the stage 1 dataset will be driven by the events that will occur in stage 2. The reason we're concerned about this is because if this happens, then we can very easily verify that the stage 1 and stage 2 test statistics will no longer be independent. The fundamental assumption used in the combination function principle will be violated, and we can no longer guarantee type 1 error rate control. The type 1 error rate may no longer be protected. And therefore, to control the type 1 error rate, the length of event follow-up in stage 1 must be fixed. There is a nice paper that discusses important considerations related to this issue. And what I would like to add here is that the maximum length of event and patient follow-up does not have to be fixed at the beginning of the study. The trial sponsor can potentially wait until the interim analysis, and then the sponsor can specify the length of additional follow-up for all patients who are included in the stage 1 dataset. For example, the sponsor may state that all stage 1 patients will be followed up for 6 months after the interim analysis, and once that trial milestone occurs, all events in the stage 1 dataset will be censored. Before we begin discussing how we can apply the closure principle and the combination function principle to help protect the overall type 1 error rate in this trial, I would like to point out that at the end of the first stage, the trial sponsor can consider a variety of decision rules. That includes a futility stopping rule, a population selection rule, and also a possibility to increase the target number of events. We can refer to this as an event count adjustment rule. Those could be tasks 1, 2, and 3 on the sponsor's to-do list. And in addition to this, the sponsor will need to think about how to develop an appropriate multiplicity adjustment that once again makes use of the closure principle and combination function approach and helps control the overall alpha at a predefined level, for example at a one-sided alpha of 0.025. And we're going to focus now on task 4. Inferences at the interim and final analyses. In this section, I'm going to walk you through the process of developing a multiplicity adjustment that controls the overall type 1 error rate in this trial across the decision points and also across all possible other sources of multiplicity. In this particular case, in example 5, the second source of multiplicity is due to the fact that there are two null hypotheses will be tested at the interim or final analysis. These hypotheses of no effect are defined on the slide. We're going to assume that H1 corresponds to the null hypothesis of no treatment effect within the overall population, which will be denoted by OP, and H2 will correspond to the hypothesis of no effect within the pre-specified biomarker positive subpopulation, denoted by M+. We need to define a multiplicity adjustment that controls the alpha level locally, so to speak, which means that it protects the type 1 rate only for this source of multiplicity, and we will then apply this multiplicity adjustment along with the combination function principle. It makes most sense in the context of this clinical trial to take advantage of the fact that we know at the design stage that the test statistics within the overall patient population and the predefined subpopulation are positively correlated. The reason we know that is because the subpopulation is nested within the overall population, and that induces positive correlation. Since the correlation is positive, we can take advantage of more powerful multiplicity adjustments known as semi-parametric tests. We have made multiple references to semi-parametric tests throughout modules C and D. And in the context of this very simple multiplicity problem, we can apply the Hagberg procedure, which would be an example of a semi-parametric procedure. As you know, as you remember, the Hagberg procedure relies on a step-up testing algorithm. And to be able to define this algorithm, we need to first define the ordered p-values. They are defined here using the standard parenthetical notation, and also the ordered hypotheses that correspond to the ordered p-values. The testing algorithm in this case is quite straightforward. There are only two null hypotheses to test, and therefore the Hagberg procedure rejects both null hypotheses if the larger of the two p-values is less than or equal to alpha. But if this is not the case, if the larger of the two p-values actually exceeds alpha, then we're going to continue to testing the hypothesis which corresponds to the smaller of the two p-values, and if that p-value is less than or equal to alpha over 2, then the corresponding hypothesis will be rejected. The next important step here will be to present the Hagberg procedure defined on slide 32 as a closed testing procedure. The closure principle, which is used for building closed testing procedures, was introduced in part 1 of this online course, and we spent some time discussing the process of setting up closed testing procedures in module D when we defined mixture-based gatekeeping procedures. We remember from module D that to be able to define a closed testing procedure, we need to go through the following 3 steps. First of all, we need to define a closed family, which includes all possible intersections of the hypothesis of interest. There are 2 null hypotheses in this multiplicity problem, and this leads to 3 possible intersections. Those intersections are presented on this slide using orange boxes. At the top of this diagram, we see the intersection of the 2 null hypotheses, which is H12, and in the bottom level, we see 2 orange boxes that correspond to the original null hypothesis. Then we need to define alpha level local tests for all intersection hypotheses. It is quite straightforward to accomplish this when we focus on the original null hypothesis, because within each one of those boxes there is no multiplicity. There's only one hypothesis left, and therefore it will be sufficient, as indicated on the slide, to simply compare the corresponding p-value to the full alpha level. Things get a bit more complicated within the box that corresponds to the intersection hypothesis, and this is where we need to apply the global SIMS test. The reason for that is because the Hogberg procedure is derived from the global Symes test, and the definition of the intersection p-value, denoted by p12, is given on the slide. We're now ready to proceed to the last step, and in the last step, we define decision rules for the null hypothesis in this multiplicity problem, and we say that to reject a null hypothesis, it is sufficient to demonstrate that all intersection hypotheses containing this particular null are rejected by their respective local tests, which means that, as I show at the bottom of the slide, we can claim a significant treatment effect in the overall population if the local tests corresponding to the intersections H12 and H1 are significant, or in other words, if those intersection hypotheses are rejected. And the same rule would apply to define a decision rule for determining a significant effect in the subpopulation in M+. We can claim a significant treatment effect within the predefined subpopulation if H12 and H2 are rejected. Now that we have defined a close testing representation of the Hogberg procedure that helps us address the first source of multiplicity due to the analysis of treatment effect within two patient populations, we need to combine this close testing representation with the two-stage design utilized in this trial. To accomplish this, we're going to use the table presented on this slide. We're going to think separately of the inferences within the stage 1 dataset and stage 2 datasets. Within each one of those datasets, we're going to compute the local p-values in the closed family associated with the original null hypothesis, which means that for each one of the orange boxes shown on slide 33, we're going to compute the p-values based on stage 1 data and stage 2 data. They will be denoted by p12, p1, and p2 within the stage 1 dataset. And when we move over to the stage 2 dataset, we will use a slightly different notation. We will use the letter Q, and therefore the corresponding local p-values will be denoted by Q12, Q1, and Q2. And the definitions of the p-values computed within the intersection hypothesis are shown below this table. They are once again based on the Symes global test. Things will definitely get somewhat complicated when we begin thinking about a simultaneous application of two rather complicated principles, and therefore I think it will be very helpful for us to try to building a diagram of the kind presented on slide 35. This diagram essentially takes the p-values from the table we have just defined and arranges them hierarchically. We see that within each stage we see a set of intersection hypotheses and associated local p-values, and our next task will be to understand at which significance levels each one of those p-values needs to be tested to help control the overall alpha. This is where the combination function principle comes in, and this principle, as we explained in the first section of this module, of course takes advantage of the alpha spanning approach. We're going to have to make certain assumptions about the amount of information available to us at the interim analysis. For simplicity, we'll assume that the information fraction at the interim analysis, denoted by t, is equal to 0.5, which means that the interim look will be taken after 50% of the total number of PFS events in this trial. We're going to choose a clinically relevant alpha spanning function. For example, we've made multiple references to the O'Brien-Fleming alpha spanning function in module E and explained that this is probably the most commonly used alpha spanning approach in group sequential trials, and this would also be true in the context of general multi-stage designs that are employed in adaptive trials. The O'Brien-Fleming spanning function enables us to define a stopping boundary, which is a set of either adjusted critical values or adjusted significance levels. If we here focus on a p-value scale, this spanning function helps us compute the adjusted significance levels that will be applied at the interim analysis and at the final analysis. Once again, we went over this process in detail when we talked about alpha spanning functions in Module E, and here I would like to mention that using the software tools that we discussed at the end of Module E, it is very easy to derive the one-sided significance levels at the interim and final. And if we assume the information fraction of 0.5, U1, the adjusted significance level at the interim, will be equal to 0.0026, and the adjusted significance level at the final analysis will be equal to U2, or 0.024. Now that we have derived the adjusted significance levels from the O'Brien spanning function, and those adjusted significance levels will be applied at the interim and final analysis, let's talk about the decision rules that will be derived for the interim analysis in this two-stage trial. Here things are going to be quite straightforward, because in this case we're looking at a decision point at the end of the first stage, and we're essentially treating it as if this was a fixed sample design. We're going to construct a diagram which is very similar to the diagram that we used when we derived a close testing representation of the Hogberg procedure. In this diagram, there are three boxes. At the top of this diagram, we see the box that corresponds to the intersection hypothesis, and within this box I have placed the intersection p-value p12. To be able to now claim a superior efficacy in the overall population, all we need to do is to show that the local p-values for the hypotheses that include H1 are significant. The adjusted significance level that needs to be applied at the interim analysis is 0.0026, and therefore we can justify a decision to stop the study early due to superior efficacy within the overall patient population if p12 is less than or equal to that adjusted significance level, and simultaneously with that, this is a very important point here, p1 is also less than u1. The same idea will be applied to derive the decision rule for the predefined subpopulation of biomarker positive patients, or M+. In this case, we need to demonstrate a significant effect within the intersection as well as within the original null hypothesis, which means that we need to demonstrate that p12 is less than or equal to u1, and at the same time p2 is less than or equal to u1. If the study was not stopped early due to superior efficacy in either patient population at the interim analysis, we're going to continue to the final analysis. And to be able to perform inferences that control the alpha at the desirable level, we will need to apply a combination function. We talked in the first section of this module that most commonly the inverse normal combination function is utilized in clinical trials, and that's exactly the approach we're going to use. The formula for the inverse normal combination function is shown on the slide, and this formula depends, as we have said before, on the predefined stage-wise weights denoted by W1 and W2, and these weights simply depend on the pre-specified information fraction. In this case, if the information fraction is equal to 0.5, then W1 will be equal to 0.5 and W2 will be equal to 0.5 as well. This diagram will help us support inferences at the final analysis and derive the decision rules for the overall population and the predefined subpopulation. We're going to assume here three different scenarios. The first one is the most optimistic scenario. Here we assume that based on predefined patient population rules, a decision was made to continue enrolling all of the patients after the interim analysis, and the sponsor is interested in evaluating treatment effects within the overall population as well as the subset. So in other words, both OP and M plus are selected at the interim analysis. In this case, when we take a look at the decision rules to be applied at the final analysis, we simply gather all of the information that was collected up to this point, and we applied the combination function to derive the p-values within each one of the boxes. Again, the top box corresponds to the intersection hypothesis, and the boxes at the bottom of this diagram, I'm referring specifically to the final analysis, correspond to the null hypothesis of no effect in the overall population, and then the hypothesis of no effect within the subpopulation. Using the closure principle, it is quite straightforward for us now to develop decision rules. As shown at the bottom of the slide, we are going to claim a significant treatment effect in the overall population if both of the p-values within the intersection as well as within H1 are significant. And when we talk about the significance level that needs to be considered here, we should go back to the selected alpha spanning function and recall that at the end of the trial, the adjusted significance level that needs to be used is given by U2, which is equal in this case to 0.024. And therefore, the treatment effect in the overall population will be declared significant if both of the conditions given at the bottom of the slide are satisfied. A similar approach will be applied to determine under what specific conditions we can claim a significant treatment effect within the subpopulation. To reject the null hypothesis of no effect within the subpopulation, it is sufficient to show that both combined p-values within the intersection as well as within H2 are significant at the appropriately adjusted significance level, and the specific conditions to reach this conclusion are given at the bottom of the slide. The scenario we have considered so far assumes that a decision was made at the interim analysis to focus on evaluating the treatment effect within the overall population and predefined subset at the final analysis. The sponsor may decide to focus only on the overall population effect at the final analysis, in which case it would be quite interesting to find out how should we define decision rules if only one patient population is selected at the interim analysis. This will be covered by the following two scenarios, and we're going to begin with scenario 2. Under scenario 2, we assume that a decision was made to only select one of the patient populations for the final analysis, and specifically the overall population was selected. How should we modify the decision rules presented on slides 42 and 43 to take this information into account? It is actually quite straightforward, and the solution is presented on this slide. Since we are no longer interested in testing the hypothesis H2 at the final analysis, then the p-value for the M plus analysis computed in stage 2, which is denoted by Q2, is set to 1. And when we look at the intersection hypothesis in stage 2, since we've only selected to focus on testing the hypothesis H1, then this intersection p-value Q12 will be replaced by Q1. Given this information, we can now combine the inferences from stage 1 and stage 2 using the combination function, and the resulting p-values are shown in the right hand side of this diagram under final analysis. Since we now know how to define the p-values for each one of the intersection hypotheses within the closed family at the final analysis, exactly the same ideas that we applied before would be applicable now, and we can now claim a significant treatment effect within the overall population if the p-value shown in the top box is significant at the adjusted significance level U2, and also the p-value that corresponds to the null hypothesis H1 is also significant at the same significance level. The next two slides present decision rules that will be applicable under scenario 3. Here we assume that based on the data available at the interim analysis, the sponsor decided to discontinue enrollment of patients within the biomarker negative subpopulation, and therefore only the M plus test will be selected for the final analysis. To take this information into account, all we have to do is to modify the definitions of the p-values computed from the stage 2 data set. As you can see on the slide, the p-value for the overall population, which is Q1, will be set to 1, and the intersection p-value Q12 will be set to Q2. Once again, there is no plan to carry out the overall population test at the interim analysis. We now have just one hypothesis left to be tested at the final analysis, and a rejection decision will be made for this hypothesis, which means that a significant treatment effect will be declared within the predefined subpopulation of biomarker positive patients if two conditions shown at the bottom of the slide are satisfied. We have seen those conditions before. Those conditions simply indicate to us that the p-value corresponding to the intersection hypothesis H1 is significant at the adjusted level, and also the p-value which corresponds to the hypothesis H2 within the biomarker positive subpopulation is also significant at the same level. We are done with the adaptive population selection design for example 5, and we are ready for a summary. In this module, we have explored a broad class of problems that trial sponsors encounter in adaptive trials that employ multi-stage designs. These designs provide a flexible alternative to traditional trial designs with a fixed sample size and a single decision point. In the context of multi-stage designs, at the end of each stage, the sponsor can apply a variety of data-driven decision rules. Adaptive designs support multiple types of adaptations. This includes, as we have discussed, treatment selection, population selection, a decision to adjust the sample size or the target number of events. In this module, we focused on example 5 that employed an adaptive population selection design, which helps facilitate the development of tailored therapies that are likely to benefit a subset of the general patient population. Repeated significance tests applied at each decision point, and also data-driven decision rules induce multiplicity, and if the trial pursues other complex clinical objectives, multiplicity adjustments that protect the overall type 1 error rate are developed, as we said, by applying the closure principle along with the combination function principle. And the last comment here, as you have noticed, there is no software implementation section in this module. This is due to the complexity and richness of available adaptive design options. The adjusted critical values that need to be applied at each decision point can be easily derived from predefined alpha spanning functions. We went over the available software tools at the end of Module E, but custom software solutions will need to be developed to support general multi-stage designs that are employed in adaptive trials.